This is the General Biology for Cardiology podcast. The overview is as follows. We'll begin with the purpose of the circulatory system. Then we'll cover vessels by comparing arteries and veins. We'll cover the mammalian circulation, go into the heart into more detail. Then we'll discuss its valves, how it's rhythmically excited, and then go into blood vessels in particular, comparing arteries, veins, and capillaries. And then we'll go into blood flow velocity and blood pressure, and then finish off with fluid exchange at capillary beds. So let's go to the first topic, the purpose of the circulatory system. Now, the circulatory system is meant to link exchange surfaces with the cells throughout your body. In the context of unicellular organisms, they can exchange materials directly with the external environment, and that's because they're small, and they can just diffuse whatever they need across their plasma membrane. But in the context of multicellular organisms, this isn't possible often. And that's because the net movement via diffusion is very, very slow. The solution is the circulatory system. In a closed circulatory system, you have three components. You have the circulatory fluid, aka blood, you have the vessels, and the pump. So what happens is the circulatory system basically connects that interstitial fluid that bathes all of your body's cells with organs that can exchange gases like in your lungs, or absorb nutrients like in your GI tract, or dispose of wastes in your urinary tract / renal. All right, next, vessels, comparing arteries and veins. So arteries and veins, they're distinguished by the direction in which they carry blood and not the oxygen content of the blood that they carry. So arteries always carry blood away from the heart, while veins always carry blood towards the heart. In the context of your superior and inferior vena cava, they are veins and they carry deoxygenated blood, while your aorta, that's an artery, and that carries oxygenated blood. Now what's tricky is that for the pulmonary veins and arteries, that flips. So for the pulmonary artery that leaves your right ventricle, that carries deoxygenated blood, while the four pulmonary veins that enter your left atrium, those carry oxygenated blood. One other term you should be familiar with are portal veins. Now, portal veins, they carry blood in between capillary beds. The most common one you'll probably come across is the hepatic portal vein, and that carries blood from the capillary beds in the digestive system to capillary beds in the liver. All right, third topic is the mammalian circulation. You have two circuits. In one circuit, which is the right side of your heart, you pump oxygen-poor blood to your lungs. In the other circuit, called the systemic circuit, this is the left side of your heart, that pumps oxygen-enriched blood to all of the capillary beds in your organs and tissues throughout your body. Now, the purpose of this double circulation or double circuit system is to be able to repressurize your blood after it's been oxygenated in the capillary beds of your lungs. So we'll dig into this further later in the podcast. But when blood goes through a capillary bed, the total cross-sectional area increases, and your blood flow velocity and your blood pressure will both drop. Now, in fish, you don't have two circuits. You have one. You have one heart. You have one atrium, one ventricle. And when that ventricle pumps blood to the gills, the capillary beds in the gills will oxygenate the blood, but then it isn't repressurized. It just travels via vessels to other capillary beds in the rest of the fish. Now, in the context of you and I, we lose some of the pressure when the blood is oxygenated in the lungs, but then it gets repressurized by the left ventricle, and then it gets sent to capillary beds throughout your body. All right, the next topic is the heart. Compared to the atria, the ventricles naturally have thicker walls because they contract more forcefully, especially the left ventricle, because that pumps blood, to the entire systemic circuit. So your left ventricle has quite a bit of thickness compared to your right ventricle. Now, although the left ventricle contracts with greater force compared to the right ventricle, it still pumps the same volume of blood as the right ventricle during each contraction. And this is why they call it an isovolumic contraction, meaning the same volume. The reason this is the case is because water or blood, they don't contract in volume upon pressure. Okay, so the volume stays the same. So even though the left ventricle is pumping harder, it's still pumping the same volume of blood. There are a few key terms you should know. The first is stroke volume. That is the amount of blood that's pumped by a single ventricle in a single contraction. The average stroke volume in a human is about 70 mLs. So if you've done research, you'll be familiar with the 50 ml conical. So each contraction of the ventricle pumps just over a 50 ml conical worth of blood. Heart rate, that's beats or contractions per minute. And when you multiply these two, you get cardiac output, which is the total volume of blood that each ventricle pumps per minute. The standard cardiac output of a human is about 5 liters per minute, and 5 liters is about equal to the total volume of blood in the human body, which is quite fascinating to consider. All right, next are the valves. I'll simplify this, and simply put, you have 2 atrioventricular valves, 2 AV valves, that lie between each atrium and ventricle, and then you have 2 semilunar valves, which are located at the 2 exits of the heart, where the pulmonary artery leaves the right ventricle and where the aorta leaves the left ventricle. Next is rhythmic excitation. So in vertebrates, the heart originates its own beat. That means that if you excise somebody's heart and just placed it in a vessel, it would keep on beating. And this is because of a group of autorhythmic cells that are located in the wall of the right atrium. Now, these cluster of cells, they're called the sinoatrial node, the SA node, and they act as a pacemaker. So impulses from the SA node, they first spread rapidly throughout the walls of the atria, and they cause both atria to contract together. Then the signal goes to the AV node, the atrioventricular node, and it's here that those impulses are delayed for about 0.1 seconds before they continue spreading towards the heart apex. This delay, it allows the atria to empty completely before the ventricles contract. Now, after that delay, the signals will continue and they'll be conducted towards the heart apex through the ventricular walls by specialized structures that are called bundle branches and Purkinje fibers. There are physiological cues that can alter your heart tempo by regulating the SA node. The first is your sympathetic and parasympathetic nervous system, the fight or flight response. And then the second is hormones, for example, epinephrine. And the third is temperature. As temperature increases, so does your heart rate. Next, blood vessels. Let's compare capillaries, arteries, and veins. So all blood vessels, they contain a central lumen, a cavity, which is lined by an endothelium, which is a single layer of flattened epithelial cells. Capillaries, they're the smallest blood vessels. They have a diameter slightly larger than that of a red blood cell. And they have very thin walls as well. It's just the endothelium, and it's surrounded by an extracellular layer called the basal lamina. So your capillaries is just the endothelium and then the basal lamina. Now, in contrast to capillaries, arteries and veins, they have 2 more layers outside of the endothelium. On the most outer, you have connective tissue, and this allows the vessels to stretch and recoil, in particular the arteries. And then after that, you have smooth muscle. So it goes connective tissue, smooth muscle, and then your endothelial layer. For arteries, the walls are thick, strong, and elastic, and this is what allows them to accommodate blood during systole when the blood pressure increases and it kind of bulges outward, and then during diastole, when the heart relaxes, it will recoil back. Veins, they naturally have less smooth muscle, and they are conveying blood back towards the heart at a lower pressure, so they don't need thick walls. Okay, but they do have one-way valves, and as your skeletal muscles contract, they will help move blood along the veins. All right, let's now move over to blood flow velocity. Because water doesn't compress under pressure, if you have water flowing through a pipe, the volume of water moving through any point of the pipe must be the same as the volume of water flowing through any other point in the pipe at any given time. This is why when the cross-sectional area of the pipe is smaller, when it decreases, say because of stenosis, or say because of an atherosclerotic plaque, the volume of water, the flow velocity will have to increase because it has to keep the volume of water passing through that smaller cross-sectional area the same. Simply put, say you have a garden hose. If you cover the nozzle with your thumb, you decrease the cross-sectional area. What happens? The flow velocity increases. Okay? So how does this relate to capillaries? The total cross-sectional area increases greatly for capillary beds compared to any other blood vessels. And because the total cross-sectional area increases, your blood flow velocity through capillaries decreases. Okay. Next is blood pressure. Once blood enters the millions of tiny arterioles and capillaries, the narrow diameter of these vessels, they create substantial resistance. Okay? And so by the time that the blood enters the veins, that resistance has dissipated much of that initial pressure that was generated by the left ventricle. Arterial blood pressure is the highest during ventricular systole, during contraction, and that's what we call the systolic pressure, which is the top number of your blood pressure, usually around 120. And during the systole, the arteries bulge out. Now, during diastole, when the heart relaxes, the arteries will snap back, and this is what you call the diastolic pressure. And that is the bottom number of your blood pressure, typically around 80. And this bulging out and contraction, this is what leads to you being able to hear a pulse or feel a pulse. Now, blood pressure can be regulated. You have nitric oxide, which is a gas, and that can induce vasodilation, while endothelin, which is a peptide, that can induce vasoconstriction. Let's finish off with the last topic, which is fluid exchange at capillary beds. So capillaries lack smooth muscle. So then how do you control blood flow in capillary beds? And this happens via two opposing forces. The first is blood pressure, and that tends to drive fluid out of the capillaries. And the second, osmotic pressure, which tends to draw fluid back in. Where does the osmotic pressure come from? Well, you have a lot of proteins in your blood that are too large to pass easily through the endothelium, so they remain in the capillaries. And these dissolved proteins, they are what are responsible for the osmotic pressure in the blood. Because there's a higher solute concentration within the capillaries compared to the interstitial fluid outside. So as a result, water wants to flow in to the capillaries, and that tendency or that pressure is what they call the osmotic pressure. So on average, blood pressure is greater than the osmotic pressure, and this leads to fluid typically flowing out of the capillaries. There's a net loss of fluid from capillaries. And in fact, each day in an adult human, there's a loss of approximately 4 to 8 liters of fluid from the capillaries into the surrounding tissues. And that lost fluid is recovered and returned to the blood via the lymphatic system. So that concludes today's podcast on general biology for cardiology.